Okay, welcome to your Kin 225 lecture. I was attempting to do some Dave Brubeck there. It probably sounds better on the CD. These are your announcements for today. Your sixth lab is due on, your seventh lab is due on April 6th. And I've got this link down here. You can't see it very well, but it's on your PowerPoint. I covered a section on aging in the course, and I've been reminded that I should also cover the other age of the age spectrum, so children and youth. So I've got a quick video link there about physical activity programs for kids. Specifically, when you go onto the link, you will have to click onto a speaker to get the interview about that physical activity in kids topic. So in today's class, I'm going to be reviewing fat metabolism, and I'm going to add a few more details on fat metabolism, specifically the effect of low and high carbohydrate feeding around exercise sessions and how that can affect fat metabolism. And then I've got an exercise for you on whole body fat metabolism after eating and during exercise. And then the last part of the lecture, I'm going to get into protein metabolism. So a few review questions here. Which has the higher lipid content, low density lipoproteins or high density lipoproteins? The correct answer here is your low density lipoproteins. So they have a low density because fats, triglycerides, or lipids have a low density. And so that's the type of lipoprotein you have if you're carrying a lot of triglyceride or a lot of lipids, is the low density lipoproteins. Where is NADH produced? The correct answer to this one is all of the above. So recall in glycolysis that you produce NADH, and if you're exercising aerobically, the hydrogens from the NADH can be taken up by the mitochondria and eventually delivered to the electron transport chain. Same as with the Krebs cycle and the beta oxidation. So Krebs cycle and beta oxidation are in the mitochondria. Krebs cycle will produce 3 NADH. Beta cycle will produce 1 NADH. Those both can go to the electron transport chain. For each NADH that goes to the electron transport chain, you're producing 3 ATPs. Which is better for stimulating fat metabolism? So consuming, say, carbohydrate like a Gatorade or sport nutrition bar before your exercise session, or doing the exercise session in a fasted state. The correct answer to this question is B, doing the exercise in a fasted state. So this is what the research has shown with regards to training in a fed versus fasted state. So if you train in a fasted state, it forces your body to use fats as a fuel source, and you'll get an upregulation of your fat oxidation pathway. So your muscles will get better at better at burning fats. Now, some studies show that it translates into better performance. Some studies show that it doesn't. One of the problems with training in a fasted state is you can't really reach high intensities if you're feeling weak because you're fasted. So one suggestion, and this is from some Australian high performance scientists, is to train with high carbohydrate intake in the evening so that you get a quality training session in, and then train in the morning in a fasted state so you get that stimulation of fat oxidation. With regards to studies on fat loss, it's shown that if you train in a fasted state, it seems to be superior for burning fats, and so you get greater fat loss. This is even if the daily caloric consumption does not change. So if you're trying to lose fat, it's probably better to train in a fasted state. So if you're, if you exercise first thing in the morning, it would be good to go out and do an aerobic session before your breakfast rather than after your breakfast. So do the aerobic session first in a fasted state, and then wait a while before you eat your first meal of the day. Now we've got this exercise where I've asked you to put these labels on these two diagrams. So one diagram has to do with fat metabolism after a meal, and the other one has to do with fat metabolism during exercise. So you can read through this slide and see the terms, and then I've got some notes on the side here that can give you some hints on where these terms might go. And what I'm going to do is shift over to this whiteboard here, and I will help you fill in the terms here. So after a meal, you have foods that are absorbed from the small intestine. In this case, with regards to fats, the important ones are your triglycerides. So that can go in this box. And the other one is also glucose. So glucose can be taken up by adipose tissue. It can be stored as fat. So once your triglycerides and your glucose are in your blood and they're trying to get into the adipose tissue, you need an enzyme here that breaks down the triglyceride to get across the membrane of the adipose tissue. So the triglyceride is too big to get across the membrane. So there's an enzyme in here called lipoprotein lipase. And that helps to break down the triglyceride into smaller components so it can get past this membrane. So it gets broken down to glycerol and fatty acids. Specifically, it's 3 fatty acids. And then those can be recombined to form triglyceride in your adipose tissue. And that's your main storage form of fat in your adipose tissue. With regards to glucose, what you need to get glucose into the adipose tissue is insulin. So insulin is a hormone that gets released after a meal. It stimulates the packing away of metabolites, so of fuel sources. So in this case, it's glucose. And within the adipose tissue, glucose can be converted to acetyl-CoA and then to fatty acids, or it can be converted to glycerol, and then those can combine to form triglyceride. So that is essentially your fat metabolism, specifically after a meal. Then you flip over the page here, you can see that things get completely disorganized during exercise. But during exercise, you're going to start off here with triglycerides. And the object during exercise is to break down your triglycerides so that you can get fuel sources for muscle. So triglycerides get broken down. Again, they're too big to get across the membrane, so they have to be broken down to smaller components. So you're going to have a glycerol and fatty acids. Now, the enzyme that breaks down the triglyceride is called hormone-sensitive lipase. I'll try to squeeze that in here. It's called hormone-sensitive lipase because it is sensitive to the effects of some hormones that are released during exercise. So those hormones would include, you're looking at growth hormone, norepinephrine, and epinephrine from your list on the previous page. So those are the hormones that help you to break down fats during exercise. Growth hormone, your norepinephrine, and your epinephrine. So those hormones are released during exercise to stimulate the hormone-sensitive lipase, That breaks down your triglyceride to glycerol and fatty acids. Now, you can see that you can also get absorption of triglycerides from the digestive tract. So you might have some triglycerides floating around in your blood from a previous meal before you exercise. Again, that can be broken down by hormone-sensitive lipase. I'm going to just put HSL for hormone-sensitive lipase at the muscle. The fatty acids can be taken up by the muscle. From there, they go into the mitochondria, where they can be broken down and used as a fuel source. You can see you have this glycerol. The glycerol cannot be taken up by muscle, but the glycerol can go to the liver. And as you know, at the liver, it can be converted into glucose. So I'm going to put glucose here, even though I don't have a box. That glucose can go back to the muscle and be taken up and used as a fuel source. So again, that's gluconeogenesis in the liver. So this is kind of the process that occurs during exercise. Okay. So I filled in those blanks. Now, before we get into protein metabolism, you'll see that I posted a crossword puzzle for you to review carbohydrate and fat metabolism. So I'm going to quickly go over that crossword puzzle with you here. Okay, so here's your crossword puzzle. You can read the clues off of the PDF that I posted. So one across, this is a metabolic pathway where most of our ATP is produced. This long answer would be your electron transport chain. And thankfully it fits. So that's your electron transport chain. The 7 across in R^1 indicates oxidation of this substrate. The answer there would be carbohydrate. Recall RQ is your carbon dioxide output divided by your oxygen consumption. If it's 1, it's carbohydrate. If it's 0.7, it means fat oxidation. Number 10 across, the number of ATP produced when FADH goes to the electron transport chain. The short answer here is 2 ATPs per FADH. 12 across, a greater amount of this organelle in muscle fibers helps to keep lactate low in aerobically trained individuals. The correct answer here would be your mitochondria. So your mitochondria can take up lactate while you're producing it, and it can oxidize the lactate and use it as a fuel source, and that will reduce your lactate levels. Okay, the main storage form of carbohydrate in muscle and liver, this would be glycogen. Glycogen. So recall that glycogen is made up of many glucose molecules. So glucose is a 6-carbon molecule, and glycogen is the storage form of the glucose in the muscle. So it's made up of many glucose molecules, the storage form of carbohydrate in muscle and also liver. 16 across, this term refers to making of new glucose, and that occurs mainly in the liver. This would be gluconeogenesis. So that would be your 16 across here, gluconeogenesis. Okay. Now the down ones, this is the storage form of fat in adipose tissue. It would be triglyceride. And as I've covered, just to review, it's made up of 3 fatty acids and a glycerol. It's called a triglyceride. 3 down, this metabolic pathway breaks down fatty acids 2 carbons at a time. Your correct answer here should be beta oxidation. So your fatty acids are long chains, contain long chains of carbon, and they get chopped down two at a time. The first carbon is called alpha, the second carbon is called beta. So if it's chopping it down two carbons at a time, it's called beta oxidation. It's a pathway in your mitochondria. This is a carrier of fatty acids across the mitochondrial membrane. This would be carnitine. And recall that there's a nutritional supplement you can take called carnitine that is purported to help with your fat metabolism. Some studies show it works, other studies show that it doesn't work so well. Number 5, excess production of this gas can cause RAR to exceed 1. So your correct answer here would be carbon dioxide. So that occurs during high intensity activity, comes from the buffering of lactic acid in the blood, and you produce this extra carbon dioxide that causes your respiratory exchange ratio to go over 1. This form of lactate dehydrogenase is located in the mitochondria. This would be your, I'm looking for, this is 6 down. This would be your heart form of lactate dehydrogenase. Okay, so there's a heart form that's located in the mitochondria, and then there's lactate dehydrogenase M form or muscle form that's located outside the mitochondria. 7 down, this hormone stimulates breakdown of muscle protein. This would be cortisol. Okay, so it's released during high stress situations, sometimes released during exercise. Let's see here. 8 down, the number of rounds of beta oxidation required to break down a 16 carbon fatty acid. So it breaks down the fatty acid 2 carbons at a time, and the number of rounds here is actually 7. It's not 8 like you might think it would be. So 16 carbon fatty acid. I'm just going to draw the carbons here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So if you broke this down 2 carbons at a time, going through beta oxidation, you'll see that it slices the carbons 1, 2, 3, 4, 5, 6, 7. So your correct answer here is 7, because you always have 2 carbons left over. So it doesn't end up being 8 slices, it ends up being 7. So that's 7 rounds of beta oxidation to break up that 16 carbon fatty acid. Then each of these 2 carbons will go into the Krebs cycle as acetyl-CoA. Okay, that was, so your answer there, 8 down is 7. Okay. 9, this is a metabolic pathway where 3 NADH, 1 FADH, and 2 carbon dioxides are produced. This would be your Krebs cycle. So I've just got Krebs here. 11 down, this is the entry point for fatty acid, carbohydrate, and amino acid metabolism into the Krebs cycle. The correct answer here is acetyl-CoA. So acetyl-CoA would be your answer. Okay, 13 down, this is the number of NADH produced during 1 round of beta oxidation. It is only 1. Okay, so you get 1 NADH produced, 1 FADH produced for each round of beta oxidation. But you can see for like a 16 carbon fatty acid, you'd have 7 rounds of beta oxidation, so you get 7 NADHs produced in this example. 14, if concentrations of this metabolite are high, metabolic pathways that break down carbohydrate will be inhibited. So the correct answer here, and this is a little bit tricky, it's ATP. So if your ATP levels are high, then it means that you are not exercising, you're probably resting, and your muscle doesn't want to go through those metabolic pathways or it's breaking down carbohydrates or fats or proteins because you don't need ATP in that situation. So ATP, if it's high, it actually inhibits those metabolic pathways that break down carbohydrates, that break down fats for energy. Okay, so that is your crossword puzzle. So the next section I'm going to get into here is on protein metabolism. You can see that proteins, just like carbohydrates and fatty acids, are made up of carbons, hydrogens, some oxygens. Unique thing about amino acids, though, from proteins. So this is the building block of protein. So a protein is made up of many amino acids. The unique thing about amino acids is they've got this NH3 group, which is called an amino group. So the key here is that you have to break down proteins if you're going to be using them as a fuel source. And they do contribute to about 5 to 10% of your energy during long duration exercise, either directly in muscle or by conversion to glucose in the liver. The key is that amine group, the NH3, has to be removed first. So it has to be removed and then it has to be gotten rid of some way, because when this is floating around in the muscle, it tends to be toxic for the muscle. So the removal of NH3 is by two mechanisms. One is called transamination. It's the exchange of that amine group between molecules, and that occurs mainly in muscle. And there's also oxidative deamination that occurs in the liver. And when that happens in the liver, the NH3 is excreted from the body. So it'll show up in your urine. Transamination, this is how proteins or amino acids can be used as a fuel source during aerobic exercise. So the NH3 can be removed, and then your amino acids can enter your Krebs cycle at different points in the Krebs cycle. So they actually keep the Krebs cycle intermediates replenished, and that will enhance your aerobic metabolism. So again, this is a process that would occur during exercise. You get some protein breakdown, especially during aerobic exercise. The amino acids from that protein breakdown can enter the Krebs cycle, but only after removal of that NH3. So this is just a summary of the importance of amino acids during aerobic exercise. You get replenishment of intermediates of the Krebs cycle, and then amino acids can also be converted to glucose in the liver. So this is gluconeogenesis. The glucose can then go back to the muscle to be used as a fuel source. One cycle I want to discuss is the glucose-alanine cycle. So this involves amino acid uptake by the liver and gluconeogenesis involving the amino acid alanine. So within your muscle, you have amino acids that are from protein breakdown. As I mentioned, you can get this transaminase enzyme working on them to remove the NH3. The amino acid can enter the Krebs cycle. Now, as I mentioned before, that NH3, when it builds up in the muscle, it tends to be toxic. And so it has to be carried out of the muscle. It combines with pyruvate, which comes from the breakdown of glucose. When you have pyruvate combining with NH3, you get this amino acid called alanine. Alanine will go into the blood, be taken up by the liver. It can be converted back to pyruvate and then back to glucose. And then that glucose can go back to muscle to be used as an energy source. And then within the liver, you get the NH3 that's taken off by this deaminase, and then it's excreted in the urine. So this metabolic pathway is very active during exercise. It allows you to break down proteins or amino acids. You get some energy by putting them into the Krebs cycle, and then you get more energy by delivering the amino acid alanine to the liver to be converted back to glucose. So if you took blood measurements during an aerobic exercise session, you will notice that your alanine levels actually get elevated in your blood because of this pathway. So this is a summary of the glucose-alanine cycle. It's activated if you're fasting, starving, or during long-duration exercise. Its purpose is to maintain blood glucose levels. So recall that your muscles taking up glucose during exercise, but your brain requires glucose as a fuel source. So your blood glucose levels are well defended, and it may provide 5 to 10% of the energy needs during prolonged exercise. Now, the next section I have here for you is on protein turnover. So proteins in your muscle, they constantly go through cycles of synthesis and then degradation. So they're constantly being made and then broken down, especially if you're adapting to exercise training. So if you're doing chronic exercise training, you know that you get increase in certain amounts of proteins in your muscles. So you get greater synthesis, you get removal of damaged proteins through degradation. Types of proteins that are important for exercise, and this is a bit of a review, are your myofibrillar proteins for strength, and your mitochondrial proteins for aerobic capacity. So within the muscle, your protein synthesis is controlled by within your nucleus by your DNA. So this is a question that asks which of the following is not found in the nucleus. The correct answer here is transfer RNA. So DNA and messenger RNA are kind of the initial first steps in protein synthesis within your muscle. Both of those are found in the nucleus, and they're important for protein synthesis. Which of the following carries individual amino acids to sites of protein synthesis in a muscle fiber? So for your protein to be synthesized in your muscle, it needs amino acids that are the building blocks for protein. And the molecule that does that is transfer RNA. So if you're confused, I've got some more explanation here. The nucleus contains DNA. Your DNA contains your genetic material that encodes for all your proteins that are made in the cell. And you can get many changes that occur at the level of your genes. So if you go on an exercise training program, there's a signal that's sent to your DNA to increase expression of certain proteins. An mRNA is a molecule that transcribes or copies a portion of the DNA and takes it out of the nucleus for protein synthesis. So your DNA encodes for all your proteins, but at a certain time, only certain proteins will need to be expressed. For example, if you're doing weightlifting exercise, you'll get increased expression of your myofibrillar proteins, your actin, your myosin, your regulatory proteins. So your DNA contains copies of those proteins. The mRNA will copy the message for those proteins, take it out of the nucleus, and then help to synthesize your proteins outside the nucleus. So I've got this process outlined in this diagram. Important components here are your nucleus, and then outside the nucleus you've got what's called a ribosome. This is essentially where protein synthesis occurs. This is where the message for protein synthesis is initiated. So within your nucleus, you have your DNA. It encodes for all your proteins. If you want to increase expression of one of those proteins, there's a message from the DNA that gets copied onto messenger RNA. So now this would contain a message for synthesis of a new protein that was encoded on your DNA. That mRNA will leave the nucleus with that message for the new protein to be synthesized. It'll go to the ribosome, and then Depending on the message on the mRNA, it's going to encode for a series of amino acids that make up a specific protein. You're going to get a molecule called transfer RNA that carries the individual amino acids to the ribosome one at a time so that your amino acid is constructed at the ribosome. So your transfer RNA carries the amino acids in there. You get this long chain of amino acids that are built on the ribosome, eventually making a new protein. And then the last stage here is after the protein, after it's served its function, it's eventually degraded, and then the process starts over again. So you get a turnover of protein synthesis and protein degradation. The process that occurs in the nucleus, so the copying of a message from DNA to messenger RNA, is called transcription. And then this process down at the ribosome where you get translation of the message on mRNA to make a new protein is called translation. So transcription and translation are important processes in protein synthesis. Your genotype describes the type of genes you have, so that's encoded in your DNA. Your phenotype describes the outward appearance of your muscle. So phenotype usually reflects your genotype. Your phenotype can change depending on, if you're exercising, if you're doing aerobic exercise or chronic strength training, your phenotype will change differently because of the expression of different proteins. Now, this question asks, what specialized type of cell may be involved in repair of muscle damage after exercise sessions? The answer here are these satellite cells. So they act to repair muscle after exercise, and they will aid with protein synthesis. So they sit outside the muscle membrane in a resting state, and these myonuclei just represent the individual nuclei. So this is one nucleus, multiple is nuclei. Myonuclei are your nuclei within a muscle fiber. So this single is a nucleus, multiple is nuclei, myonuclei specifies muscle fiber. So again, this is where protein synthesis initiates. With exercise training, especially with resistance training, and this is before resistance training, this is after, you see two things happening. You get replication of the satellite cells, and the satellite cells also get incorporated as new myonuclei. So recall, this is where protein synthesis starts in the nucleus. And so if you have more of these, you have greater capacity for protein synthesis. So control of these satellite cells is very important for controlling protein synthesis. There are your satellite cells. So two things happen with exercise training, especially resistance training. You get multiplication of the satellite cells, so proliferation. And then you get the satellite cells turning into myonuclei, and that's called differentiation. So both occur to satellite cells with resistance training. So this is a summary. Resistance training increases the number of satellite cells and myonuclei. The satellite cells get incorporated into muscle fibers as new myonuclei, and this results in increased capacity for protein synthesis. Last question I have for you here is, two individuals of the same age and sex will respond the same if presented with the same resistance training program. You probably know that this is false. So individuals respond very differently to the same program. A lot of it has to do with genetics. So you get responders, non-responders to resistance training. And it's been shown that people that have a greater number of satellite cells before a resistance training program will be the responders. They will hypertrophy more or get increases in muscle size more easily with the resistance training program. And that is pretty much it for today.